Good afternoon. I am here to present my findings on proteomics of macular hole, Piwil1 as a potential regulator of retinal integrity. This work was done along with our collaborators from Department of Biochemistry and Molecular Biology at the University of Pondicherry. Let us have a quick overview of molecular biology. Inside the cell nucleus, the human DNA undergoes the process of transcription to give rise to the RNA. The messenger RNA codes for structural proteins, while the non-coding RNA performs various regulatory functions. One of these is the piRNA, which binds with the PV proteins, which further bind with different proteins in the cell to carry out various regulatory functions or influence their action. So piRNAs are small non-coding RNAs. They bind with PV proteins as called in Drosophila, and PV-like proteins in other species are known to silence transposons, are present in somatic cells, and were first time identified in ocular tissues by our collaborators. And they have shown dysregulated expression of Piwil4 to be linked to retinoblastoma and diabetic retinopathy. So we propose in this study to investigate the potential role of Piwil1, a member of the PV family of proteins, in the pathogenesis of macular hole. IEC approved this prospective study. Undilated vitreous samples were collected intraoperatively from macular hole patients. Piwil1 expression was confirmed by immunoblotting, and protein interactions identified by liquid chromatography mass spectrometry. So the vitreous samples were cleared of debris, 10 samples pulled together, and these were then incubated with the Piwil1 antibody and the IgG antibody, which serves as a negative control. This was then subjected to electrophoresis. The gel was stained with Kumasi brilliant blue. The protein bands were excised, de-stained, and then they underwent lysis to extract the protein. This was dried and stored at -20 degrees. This was then analyzed by liquid chromatography mass spectrometry to get the output or the resultant interactions. The Piwil1 immunoprecipitation was validated by Western blot. And following the mass spectrometry analysis results, one of the interesting factors, the pigment epithelium derived factor, was again immunoprecipitated with Piwil1 and subjected to Western blot to validate our results. So we saw that the Piwil1 showed a very interesting interactome and had many novel binding factors which influence various functions in the ocular tissues. Samples from SFIL served as a control and had a distinct different interactome. So in the Piwil1 proteome interactome, we saw neuronal proteins. Calcitonin and spondin were enriched hits, which are crucial for axonal branching, compartmentalization in neurogenesis, and they support nerve growth and repair. We saw cytoskeleton organization elements which are involved in neuronal repair. We identified anti-angiogenesis factor like pigment epithelium derived factor and opticin, which regulate disease-related angiogenesis and enhance neuronal survival during stress. Calcium signaling elements, which are essential for neuronal transmission and crucial in axonal guidance and growth. Elements involved in protein trafficking, which regulate protein degradation, synthesis. Apoptosis-related factors that maintain mitochondrial function, energy production, prevent unnecessary cell death, and regulate cell growth, division, and apoptosis. And factors involved in chromatin regulation, which balance transcription, help in identifying cell identity, regulate retinal development by controlling gene expression. So to conclude, Piwil1 shows interaction with proteins that influence nerve growth and repair and neuronal transmission. These interactions possibly indicate recruitment of a neuronal repair mechanism in response to the tangential and anterior posterior tractional forces that lead to the formation of a macular hole. Piwil1 promotes neuronal recovery by aiding regeneration after injury or stress and could be a potential therapeutic target for management of macular hole. Thank you.
Yes, Dr. Bhatia.
Really excellent work, but I just want to know that in clinical practice, where do we stand with this, or where do you hope that this would go ultimately?
Yeah, so this is like a very, very preliminary kind of a biochemical analysis where we have identified that these factors are showing interaction with the Piwil1 in patients with macular hole. So probably further exploration of this and being able to elucidate how their role goes, maybe in the future we could have some kind of an agent which used either intraoperatively or maybe like an injection, where you can recruit these particular things and their interactions to help in healing. But this is a very preliminary finding and study.
Manavi, very nice study. I just wanted to ask you, did you check on any inflammatory mediators?
There were no inflammatory mediators in this, ma'am. I'm actually presenting one part of the study. The other part was the diabetic retinopathy. That's a poster over here. In those, we had inflammatory mediators, but we did not have inflammatory mediators here.
Thank you.